look for the global alignment, and then we look for the local alignment. So global alignment attempts to align the sequences across their entire lengths, means from the end, beginning to the end of the sequence, we align the entire sequence one after another, and we check which region is shared, which region is common, which region is different, where the nucleotide is different, where it lacks the nucleotide. That kind of study we do in the global alignment. In local alignment, we search for the best matching region or regions within the sequences. So here you can see, now lookly close the sequence between these two sequences, TGCTA, GCTA. Now if you look TGCTA, GCTA, and this sequence TGCTA, GCTA, these two sequences, this from these two sequences, this part of sequence is common. This part of sequence is shared. So this kind of thing we do in the local alignment. Now we'll talk about what is the fundamental difference between global versus local alignment. So here feature wise, we'll go. Main idea behind the global alignment is align the entire sequence, and in local alignment we find the best matching regions. Coverage, usually entire sequence we look in the global alignment, but in local alignment we just cover, or we try to cover only a region. It is a best for similar sequences of comparable length, and local alignment is best for sequences sharing specific regions. Global alignment focus on overall similarity, local alignment focus on local similarity. Global alignment we apply in comparing related full length sequences, and local alignment we use for searching databases for similar regions. Now, when should global alignment be used? Global alignment is particularly appropriate when two sequences are expected to be homologous across most of their length. Their lengths are relatively similar. The objective is to compare overall sequence differences, studying closely related genes or proteins. Now, example here is we have if we compare the human hemoglobin alpha with chimpanzee hemoglobin alpha. If the sequences are highly similar and comparable in length, global alignment can provide a useful overall comparison. So, if your sequence is similar, you know the sequence, your sequence belong to this protein, your sequence belong to this species, and you want to check against the sequence of another species of the same protein, then you can use the global alignment. Now, when should local alignment be used? So local alignment is particularly useful when only part of the sequences may be related. Okay, sequences have a different length. Conserved domains are being searched for. A sequence may contain a shared functional region. Searching a large database for related sequence regions. Now, where we can use it? See, when I am giving one example, an unknown protein may contain a conserved enzyme domain while having additional regions that are unrelated. Okay. Local alignment can identify the conserved domains. Suppose we know the one enzyme. Okay, one enzyme contains the sequence which is common, so we can find out what are the other domains are conserved in various species. Or suppose if you don't know about which kind of sequence it is, So we can do the local alignment and we can search against inter database to find out which kind of this um, sequence is. Okay, so that is the main idea behind the local alignment. Critical concept, see, now here global doesn't mean always better or local doesn't mean always better. Okay, the correct method depends on the biological question. Firstly, we have to ask the question to ourselves is, do the sequences need to be compared across their entire lengths, or is the important similarity expected to occur only in a region? Okay, so we'll check whether the sequences we need to check to the entire length, or just we have to find the similarity in particular region or in a specific region. So that question should come before choosing an alignment approach. Okay, so we can um, ask this question, we can study this question, and we can decide which alignment we should use. So this is the module 3.1. Okay, in the next module we'll learn about the gaps or the some fundamental things, some calculation things before proceeding with the blast and multiple sequence alignment. Till then, thank you. If you have any doubt, you can raise the question in Google form that is shared with you, or you can directly get connected with me over the WhatsApp or over the call. No problem in that. Okay, just watch the entire video, and please subscribe to the channel, give the like to this video, and we'll meet in the next module. Till then, thank you.